Hello students, today we can see the topic enzymes. What are enzymes? Enzymes are organic compounds that are produced by the living cells to speed up the biochemical reactions that take place inside the cell. The process of speeding up of chemical reaction is called catalysis, and the substance that speed up the reactions are called as catalyst. As these enzymes are secreted by the living cells and as they speed up the chemical reactions, they are known as biocatalysts. Enzymes can also be defined as enzymes are catalysts of biological origin which accelerate the various cellular reactions without themselves undergoing any apparent change during the course of action and that are not dependent on the cell for their activity. So enzymes are biocatalyst and the term enzyme was coined by Kuhn. All enzymes are proteins, but there are some nucleic acids that behave like enzymes and they are called as ribozymes. Now when we see the structure of these proteins, that is the enzymes which are proteins, they are also having the primary structure, secondary structure and tertiary structure. You all know the proteins are having primary, secondary and tertiary structure. And here the enzymes are all proteins and all enzymes are proteins. So they also show primary, secondary and tertiary structure. Okay, primary structure will be in linear form. It tells us which is the first amino acid, second amino acid or the last amino acids like that. And the secondary structure will be folded and tertiary structure will be woolen ball like a structure. So if you see the tertiary structure of the enzyme, the protein chain will be folding upon itself and this chain will be crisscrossing itself and thus many crevices or pockets are made. One such pocket is termed as the active site. So what is an active site? An active site of an enzyme is a crevice or pocket into which the substrate fits. Okay, so the enzymes are having the active site in which the substrate will be fitting onto it. So here in this picture you can see the active site is there and in that active site the substrate is fixing, the yellow colored substrate fixes into the active site of the protein, that is the enzyme. Okay. Enzyme catalysts are produced inside the living cell, isn't it? There are various inorganic catalysts also. Now we can see a few differences between enzyme catalyst and inorganic catalyst. Enzyme catalysts are generally globular proteins, but inorganic catalysts are mineral ions or other small molecules. And enzymes can promote a specific reaction of a substrate, but inorganic catalysts promote diverse reactions. Several reactions can be promoted by inorganic catalyst. Enzymes are regulated by specific molecules and no such specific regulator molecules are there in inorganic catalyst. Enzymes are synthesized by the ribosomes in the living cells. As you all know, in the rough endoplasmic reticulum, ribosomes are there. Rough endoplasmic reticulum is used for the synthesis of proteins, isn't it? So that ribosomes which are present either in the rough endoplasmic reticulum or in the cell cytoplasm, they are involved in the synthesis of enzymes. Okay, and inorganic catalysts are not synthesized in the living cells. Now, when we see about the enzymes are more sensitive to temperature and pH. We can say enzymes get damaged at a high temperature. If the temperature is very high, the functioning of the enzyme cannot take place. Why? Because they get denatured. Okay, their structure changes. So they cannot function. But there are some organisms which live in thermal vents where the temperature is very high. Even it can go up to 80 to 90 degrees Celsius or even 100 degrees Celsius. So there certain bacteria are there. These are thermophilic organisms, already you have studied. So such bacteria can withstand high temperature. So in such bacteria, their enzymes can withstand very high temperature. generally the enzymes cannot withstand such high temperature or even high pressure or different pH. Okay, so they are more sensitive to temperature and pH. But if you take inorganic catalyst, they are less sensitive to temperature and pH. They can work efficiently even at high temperature and high pressures. Okay, so this is one of the very important difference between enzyme and inorganic catalyst. That is, inorganic catalyst can work efficiently at high temperature and high pressure, but enzymes get damaged at a high temperature or even at a high pressure. Okay, so this is the difference between enzyme catalyst and inorganic catalyst. Chemical compounds undergo two type of changes. What are the two type of changes? Physical change and chemical change. What is physical change? Physical change refers to a change in shape without breaking of bonds. So it is a physical process. Change of state of matter is a physical process. For example, when ice is melting into water or when water becomes vapor, these are all change in the state of matter, isn't it? So this is an example of physical process. But on the other hand, when bonds are broken and new bonds are formed during transformation, then it is said to be as a chemical change or a chemical reaction. For example, if you see this equation, that is barium hydroxide plus sulfuric acid gives barium sulfate plus water. Okay, in order to balance the equation, we can write it to H2O. So barium hydroxide plus sulfuric acid. So the bonds which are present in them are broken and now new bond is formed. How? By combining barium and sulfate. So how it is formed? Barium hydroxide plus sulfuric acid forms a barium sulfate plus water. So this is an inorganic chemical reaction. Like this, hydrolysis of starch into glucose is an example for organic chemical reaction. Okay. Now, the rate of reaction, that is rate of physical or chemical process, is the amount of product formed per unit time. Okay, so rate of a physical or chemical process refers to the amount of product formed per unit time. And how it is expressed? It is expressed as rate of reaction is equal to delta P by delta T. And this rate of reaction can also be called as velocity if the direction is specified. Okay, that is if the direction, the reaction in which direction it is taking place, if it is specified, then we can consider it as a velocity. Now the rates of physical or chemical processes are influenced by various factors, and one of the important factor that influence the rate of reaction is temperature. A general rule of thumb states that the rate of reaction doubles or decreases by half for every 10 degrees Celsius change in either direction. That is, the rate of reaction can double or it can decrease by half when there is a change in 10 degrees Celsius in either direction. When 10 degrees Celsius change is temperature is increasing or decreasing, accordingly the rate of reaction will double or decrease by half. Okay. Catalyzed reactions proceed at higher rates than that of uncatalyzed ones. So when we add enzyme, when enzymes are there, the reaction can take place at a faster rate than the uncatalyzed reaction. For example, if you see this reaction given here, carbon dioxide plus water gives carbonic acid, and here the enzyme which catalyzes this reaction is carbonic anhydrase. So in the absence of this carbonic anhydrase, only 200 molecules of carbonic acid can be formed in 1 hour. Okay, but on the other hand, when this enzyme is involved, this carbonic anhydrase enzyme, it is present in cytoplasm, and when this carbonic anhydrase enzyme is involved in this reaction, the same reaction, the reaction speed will be increasing tremendously, and within a second, for every second, 6 lakh molecules of carbonic acid can be formed. You see, you compare it, only 200 molecules of carbonic acid can be formed in 1 hour in the absence of enzyme, but in the presence of the enzyme, 6 lakh molecules of carbonic acid is formed for every second. So how much acceleration is done by the enzyme, isn't it? So here it is 10 million times it has accelerated the reaction. The enzyme carbonic anhydrase has accelerated the reaction. Okay. So the power of the enzyme is incredible, isn't it? Now, there are thousands of types of reactions that take place inside our cell, isn't it? And some reactions are having a single step, but many reactions are multi-step reactions. Okay. Now, what is a metabolic pathway? A multi-step chemical reaction when each of the steps is catalyzed by the same enzyme complex or different enzymes, it is said to be as metabolic pathway. So metabolic pathway means when many steps are involved in that reaction, a multi-step chemical reaction when each of the steps is catalyzed by the same enzyme complex or different enzyme is called a metabolic pathway. For example, if you take glucose is converted into 2 molecules of pyruvic acid. Okay, so it is a metabolic pathway. So when we say simply, we can say glucose is converted into pyruvic acid, but actually it is a 10-stepped reaction. 10 different steps are there which are enhanced by 10 different enzymes. Okay, so it is considered to be a metabolic pathway. Okay, so already in 10th class we have studied glucose will be converted into pyruvic acid in the presence of oxygen. More amount of energy will be released from the pyruvic acid, isn't it? Now, so glucose is converted into pyruvic acid generally in our cells. Okay, in normal conditions, it will happen. Glucose will be converted into pyruvic acid. Now, in under anaerobic conditions, suppose if you take our skeletal muscles, when they continuously work, most of the oxygen present in them will be used up. So, under anaerobic condition or when there is lack of oxygen, then lactic acid will be formed. Okay, the same way, if you take yeast during fermentation, what will be formed from glucose? We can say ethanol will be formed. Okay, the same pathway can lead to the formation of ethanol. Okay, so in at different conditions, different products can also be formed by the same metabolic pathways. Okay. How the enzymes can bring about high rates of chemical conversions. See, the chemicals will be converted into some other chemical, or one metabolite will be converted into another metabolite, isn't it? So the chemical or metabolite conversion can be referred to as a reaction. The chemicals which are converted into a product is called the substrate. So the substrate will be converted into a product, and the substrate will be attaching to the active site of the enzyme. As a result, the substrate S can be converted into the product P. Symbolically, we can represent S gives rise to product. Okay, S is converted to product. Okay, so when the substrate attaches with the active site, what is that active site? It is the crevice or the pocket which is present in the enzyme. So when the substrate attaches to the active site, the substrate will be diffusing towards the active site, and thus it forms an enzyme substrate complex. Okay, so this complex cannot remain as such. So we can say this complex formation is a transient phenomenon. It will be changing and it will be converted into a product. Okay. So during this phenomenon, what happens? The substrate will be forming a new structure. Okay. And that structure we can say it as it reaches a transition state. Okay. Or the new structure which is formed is said to be as a transition state structure. What is meant by transition state structure? This is an unstable structure which is short-lived, and it is having partial bonds in them, and it is having a high energy level. Okay, transition state is a high energy state which is unstable in nature. So that substance cannot remain as such long for a long time, isn't it? That's why it is short lived, because it is having high energy and it is unstable in nature. So what happens? Very soon bond breaking and new bond formation takes place, and when it is completed, the substrate will now be converted into a product. Okay, and when the substrate is converted into a product, it cannot fit into the active site no longer. So what will happen? It will be released from the active site. Okay, so now the substrate is transformed into the product. So during the pathway of its transformation, that is when a substrate is converted into the product, it will be undergoing the transition state structure, which will be having different altered structural states in them. Okay, we cannot say immediately the substrate will be converted into a product. In between that, various intermediate structural states will be formed, and at last a stable product will be formed. All the intermediate structural states are unstable because they will be having high energy and they are short lived and they are unstable. Okay, and when it becomes a product, it will be stable. So the stability is something related to energy status, isn't it? The transition state will be having high energy state, but the product will be having a low energy state. So it becomes a stable product. Okay. This is a graph that shows the concept of activation energy. In this graph, the y-axis represents the potential energy and the x-axis shows the progress of reaction. Okay, it shows the progress of the structural transformation of the substrate passing through the transition state and thus forming the product. So here the product is formed, isn't it? So here, if you see the energy level difference between S and P, the substrate and product, the product is having low energy level, isn't it? So if the product is lower than that of S, then we can say it as an exothermic reaction. So in exothermic reaction, we no need to supply energy by heating to form the product. Okay, so whether it is exothermic or endothermic reaction, the substrate has to go through the transition state to reach the product. Okay, the substrate S has to go through the transition state, then only it can reach or it can be converted into a product. So what is that transition state? I already told it is the high energy state, isn't it? So what is required to provide energy? Activation energy should be provided to the substrate so that it can enter into the transition state and then it can be converted into the product. So what is this activation energy? It is the difference in average energy content of substrate S from that of its transition state is called the activation energy. Okay, the difference between the energy content in substrate S as well as the transition state is said to be as what? Activation energy. Okay, so the substrate S has to take or has to get more amount of energy from its surrounding. That energy is the activation energy. It should get it from the surrounding so that it can reach a high energy state, that is the transition state. Okay, then automatically it can be converted into the product. Okay, but if we are adding an enzyme, this enzyme can bring down the energy, activation energy amount required for the conversion of substrate S to the transition state. So here if you see in this graph, you can see here this is the activation energy with the enzyme. So very little amount of energy is required. But here it is activation energy without enzyme. So the amount of activation energy required for converting substrate S to its transition state is more. Okay, so energy activation energy amount can be brought down when enzymes are added to the reaction. Okay. Now how the enzyme action takes place. Each substrate S will be binding to the enzyme E in its active site, isn't it? As a result, an enzyme substrate complex is formed. That is, E plus S gives ES. ES is enzyme substrate complex is formed. Now what happens to this complex? It is short-lived. Immediately it will be forming enzyme product complex. Okay, that is also short-lived. Then immediately it will be dissociating into enzyme and the product. Okay. So the nature of enzyme action can be explained under 4 steps. What are the 4 steps? The first step is the substrate binds to the active site of the enzyme, fitting into the active site. Okay. Only to the active site the substrate will be attaching. To other region it cannot form bond and it cannot attach. Okay. Then second step is the binding of the substrate induces the enzyme to alter its shape. It will be changing its shape, so that the enzyme will be changing its shape so that it can fit more tightly around the substrate. Then third is the active site of the enzyme, which is in close contact with the substrate, breaks the chemical bond of the substrate and helps in the formation of enzyme product complex. Okay, the bonds will be broken and new bonds will be formed and an enzyme product complex is formed. Then the last step is the enzyme releases the product, because enzyme product complex is formed. Now the product, when it is fully formed, it releases the product. The enzyme releases the product of the reaction and the free enzyme is ready to bind with another molecule of the substrate and again it can help in another catalytic cycle. Okay, so this is how the enzyme action takes place. This is the diagrammatic representation of the enzyme action. Here you can see the active site of the enzyme to which the substrate will be attaching and it forms the enzyme product complex, and when the product is formed it will be released out and the enzyme will be free so that it can bind with the another substrate. Okay, that is about the enzyme action.